today we're finishing the replication cycle. We've gone through every step in some detail, and you now know how viruses replicate, and today we're going to learn how to make new viruses. And then Wednesday we're going to start talking about infection of multicellular organisms. So far we've been talking about infection of cells, single cells in culture, but now we're going to do animals, pathogenesis, for the rest of the course. But let's build some viruses today and see what we can come up with. If you look at a virus particle, you should, by the end of this lecture, be able to say exactly how it's assembled. And here are some of the viruses we've talked about so far in this course. They include viruses with capsids that are non-enveloped, viruses with envelopes with and without capsids. And today you'll be able to figure out, if I showed you a picture of one of these, like that one there on the left side, which is influenza virus, you'll be able to figure out, or at least give me in broad strokes, how it's assembled. Now, each virus has to go through a series of assembly steps in order to be put together. So we're now at the infectious cycle. We've made proteins, we've made genomes, and we are ready to put them together and make a virus particle. So here are some general steps that we have to go through, and they're color-coded depending on whether they are common to all viruses in yellow, or only some viruses do these particular steps in blue. So we start out by formation of structural units of the protein shell, and then we assemble the shell by interactions among structural units. And we go back to our lecture on structure to understand the principles by which protein subunits come together and make either helical or icosahedral structures. And then the third part, we have to package the nucleic acid. This is a very important part, obviously. If you make a beautiful coat but forget to put nucleic acid in, it's of no use. The infection's not going to go anywhere. On the other hand, you have to make sure you don't package non-viral nucleic acids in the particle. That would be a waste because they don't do you any good either. So you have to have selective packaging. We're going to talk about how that happens today. Now, for some viruses, you need to get an envelope, a membrane, a lipid membrane, which of course comes from the host cell. And not all viruses obviously do that, so that's why it's colored blue here. But all viruses have to be released from the cell. At least most of them do. There's always exceptions to everything in biology, and there are actually some viruses of yeasts and fungi that never leave their cells. They form these beautiful particles and they stay within the host cell. But for the most part, the viruses we talk about have to be released. Why would they have to be released? The only way you can spread to another organism is to get out of the first one. And that's that part. And finally, there's one more blue step, maturation of virus particles. Some viruses actually mature after they've been released from the cells. Not all of them, but some of them do. We'll actually talk about one of them today. And that's why it's in blue, and it's after the release from the host cell. You would think, well, if you're being released, you're going to be mature. But in fact, there are some that aren't completely formed until after they leave the host cell. Just like every other step of virus replication, assembly is dependent on the host cell machinery in many, many ways. It's not just a matter of making a protein and it assembles spontaneously into a virus particle, although that in some cases can happen. And when we talk about vaccines, we'll talk about vaccines which you can make by producing a single viral protein and it spontaneously assembles into a capsid. But that's one of the exceptions. Most viruses need other things from the host cell. This includes chaperones, cellular proteins, whose job it is to make sure that the viral proteins fold properly and interact properly, and if they are misfolded, to get them out of the picture. Transport systems. We've talked about how viruses infect cells. They have to come from the plasma membrane all the way to the nucleus sometimes. They don't just diffuse. Remember, they use transport systems of the host cell. On the way out, once you make virus particles, let's say you make them in the nucleus, they're not going to just diffuse out. They use transport systems to get up to the plasma membrane. And many viruses use the secretory pathway. That's the pathway by which a protein is made in the endoplasmic reticulum and is shunted up to the plasma membrane. And we'll see today how budding viruses, envelope viruses, depend on that pathway. And of course, nuclear import and export is also important because many viruses have to get in the nucleus in order to replicate their genomes and in fact to assemble, as you'll see today. And then once they're there, they have to get out. So we need import and export signals. Again, I want to emphasize this point that you just don't diffuse in cells. We've talked about this before, how it would take too long for a virus to go from the plasma membrane to the nucleus. And so the virus particles are carried on specific transport pathways. And on the left part of this slide, we're looking at two specific kinds of transport pathways. One which we really haven't mentioned very much, but short distance energy-dependent protein channels are often needed to move virus particles, say, into cells. But more important for our discussion today, long-distance movement depends on energy-dependent motor proteins, such as the microtubule network, which is illustrated in a cartoon fashion on the right here, to get not only from the outside to the inside of the cell, but again, after being assembled, to move from the nucleus to the plasma membrane. And on the right is an experiment that illustrates this and makes it very clear. This is a cell infected with vesicular stomatitis virus. And on the top is an untreated infected cell. And it's stained with a few things. First, the virus particles, the nucleocapsid, is stained yellow. The nucleus is stained blue, staining the DNA there. And the microtubule network of the cell is stained red. So you can see this extensive network. And of course, it's on the network of microtubules where the motor proteins lie that bring viral proteins and structures in and out of the cell. So you can see what happens with the vesicular stomatitis virus is that the nucleocapsids, the RNA plus protein, is assembled in the cytoplasm, and then it has to make its way to the plasma membrane. So you can see on the upper left lots of cytoplasmic distribution, but then you can see the pinpoint sites of nucleocapsid moving towards the plasma membrane. And there they will acquire a membrane, as you'll see later today. On the bottom, we have treated these cells with a drug that disrupts microtubules. It's called nocatazole, for those who are interested, but you don't need to know that. It disrupts their function. It breaks them up and makes it not possible for things to move along them. So you can see now the nucleocapsids of vesicular stomatitis virus are just accumulating in sites within the cytoplasm. They're not moving where they need to be. They're not moving to the plasma membrane because there are no microtubules left for them to move. Now, this cell will not make any infectious virus because these nucleocapsids cannot move to the plasma membrane, which is where they need to go to acquire a membrane. Okay, so just emphasizing that you need movement on motor tracks in the cell for viral proteins to move around. So that's one generality that we can apply to assembly or principle, as I like to say. A second one is on this slide. Nothing happens fast in dilute solutions, and viruses make things concentrated so that assembly reactions go quickly. And in fact, we've talked a little bit about how this applies to RNA synthesis. I told you that many RNA viruses synthesize their genomes on the surfaces of membranous vesicles to concentrate the components. And in fact, on the lower left slide is an electron micrograph of a polio-infected cell. And you can see lots of membranous vesicle, the spherical structures there, and it's on their surface. These are induced by infection. They're not there in the uninfected cell, and on their surface, RNA synthesis occurs. Well, it also turns out that the assembly occurs on the surface of these vesicles as well, and that is a good way to concentrate the proteins you need to drive these reactions forward. Otherwise, the assembly reactions occur too slowly. So that's one way, concentrate proteins on internal membranes. And the other one is shown on the right. This is a photograph of a cell by light microscopy as opposed to EM on the left, infected with rabies virus. And these cells are typically full of what are called Negri bodies, and those are those dark circles there. And what they are sites of assembly. We call them factories or inclusions, but they are where the virus particles are being assembled. So the genomes and the proteins are brought to a place, and there's so many in one place that you can see them accumulating. And Negri was an Italian pathologist living many years ago, and he was looking under the microscope and he saw these And he said, these happen in a rabies infection. He called them negri bodies, and he had no clue what they were. Now we know that they are factories for the assembly of rabies virus. And again, the reason why they exist is to speed up assembly. And these are just two examples. There are many, many more. In many virus-infected cells, you see accumulation of structures in the course of infection, and these are sites of virus assembly. So the point here is that assembly doesn't just happen everywhere throughout the cytoplasm. It happens at very discrete foci to make the reaction very efficient. Now, to get proteins, viral proteins, where you need them to be for assembly, they have addresses. And if you've taken biology, you know about such addresses to target them to different sites. For example, if you want to target proteins to membranes, you can have a signal sequence to initially put it through the membrane of the ER. You can have fatty acid modifications that target it to other membranes, and we'll see some examples of this today. There are membrane retention signals that keep proteins in the ER. For example, nuclear localization signals, or NLSs, that allow a protein to get in the nucleus by the nuclear transport machinery, and nuclear export signals. And on the bottom of this is an illustration of two different kinds of nuclear localization signals. These are relatively straightforward sequences of amino acids. You can see a simple one here consists of hydrophobic 3 to 7 basic and then hydrophobic amino acids. And you can take this, which is in the capsid protein of SV40, in the T antigen of SV40, you can put this into another protein and it will allow that protein to get into the nucleus. That's what we mean by an address. It specifies nuclear import. So let's see how this works in a virus infection. Here are some examples of nuclear import. So we have influenza virus, which is unusual for an RNA virus in that it actually requires the nucleus for replication. Its nucleoprotein, the NP protein, is what coats the viral genome, of course, to make it a nucleocapsid. That's made in the cytoplasm, of course, as all proteins are, and it has to get in the nucleus to combine with the viral RNA. So it has a nuclear localization signal that imports it through the nuclear pore. Here we also have the SV40 capsid subunit. You can see one of the pentamers that's formed in the cytoplasm. This virus assembles in the nucleus because that's where the DNA genome is replicating. In fact, most of the DNA viruses we talk about, they replicate their genomes in the nucleus, and that's where they assemble, with the exception, of course, of pox viruses, which do it in the cytoplasm. So the SV40 capsid has to be imported. It has an NLS, similar to the one I just showed you. Adenoviruses also assemble in the nucleus after DNA replication. So all these proteins, whether it be nucleocapsid or structural proteins, have signals on them that allow nuclear import. Targeting to plasma membrane. Many viruses acquire envelopes at the plasma membrane, as we will see in a bit. And these proteins, these viral glycoproteins, have to have signals that allow them to get there. And they have a number of signals, including a signal sequence. So they are translated by ribosomes bound to the endoplasmic reticulum. The protein goes into the lumen of the ER, and then it's moved through the Golgi stacks up to the plasma membrane by a series of transport vesicles. This is the so-called secretory pathway. So vesicles bud off of the ER containing the glycoprotein. They then fuse through the Golgi stacks, and then finally the last vesicle buds off of the last stack of the Golgi and moves up to the plasma membrane. And of course, that happens on a microtubule. These vesicles are not simply diffusing. And then the vesicle fuses at the plasma membrane, and now we have viral glycoproteins embedded in the plasma membrane. And we'll see the purpose of that in a bit. Another concept that we see over and over again in assembly is the concept of an assembly line or making a sub-assembly. Now, you all know how to make cars. You know, you start with a frame and you put a door on it, it moves down to the next station, and you put a dashboard and it goes down to the next station and so forth. Used to be in the old days, this was done by people. Now it's all automated. And here it's the same thing. You make sub-assemblies. It doesn't all come together at once. And the example shown in this picture is the assembly of a tailed bacteriophage, which has an icosahedral capsid, which has the viral DNA in it, and a helical tail and tail fibers. These are made separately and then put together in a series of events that people have worked out for many viruses. So you can see the tail is composed of all these numbers are each an individual protein that goes to make up the tail. You make the base plate first, then you make the helical neck and you wrap discs around it, and now finally you have the tail assembly. At the same time, the head is being assembled as an icosahedron, and that gets then joined to the tail, and finally the fibers are assembled separately and put on last. So that's what I mean by sub-assemblies. You make different parts of the particle and then put them together. So they're distinct intermediates, as you can see from this picture, and one of the advantages of this is it gives you quality control. You can't proceed to the next step unless the whole structure is right. So if the tail you make here isn't quite right, it's not going to fit in with the capsid, and assembly will stop there. So you end up not making a lot of defective particles, okay? So there are two important things here, the concept of sub-assemblies and the idea that one of the advantages of that gives you quality control measures. Of course, I think it's the same way in automobile assembly. If you get to a certain point and something's wrong, they then finish that and send it to New Jersey so people there buy it instead of taking it out of the assembly line. Let's look at how you make a sub-assembly in some detail. I've shown you a picture using a bacteriophage. Let's look at a couple of examples using the viruses that we have talked about. We're going to look at three strategies. First, assembly from individual protein molecules. The top example is with SV40. So on the right is the pentamer. It's composed of 5 copies of VP1 and 1 copy of VP2/3 in olive color. And these are simply translated from individual mRNAs. They're produced as individual proteins. You see a single VP1 and a VP2/3. And then they're assembled into a pentamer. 5 copies of VP1 come together and 1 of VP2/3. Now, this doesn't just magically happen. These proteins have within their sequence and structures the instructions for self-assembly. And as you'll see in some cases, chaperones help this to proceed in an accurate way. But this is something that's built into the primary sequence of the protein. On the bottom is simply another example of this assembly from individual protein molecules. We're going to make the penton of adenovirus. This is consisting of the fiber and the penton base, which exists at each 5-fold axis of the virus particle. And that's made up by making a molecule of protein 4, that's a monomer. Then 3 of these come together to make the fiber, which is a trimer. And then you make a molecule of protein 3. 5 of those come together to make a penton base. Then these 2 assemblies come together. All right, so you can see we're doing assembly line, sub-assembly procedures here, but you can see where the proteins are coming from. In this case, from individual protein molecules that are separately made and then put together. Now, that's not the only way to do things, of course. There are many other ways. Second kind is assembly from a polyprotein precursor. Last time we talked about how some viruses where the genome is an mRNA, one of the strategies for making a lot of proteins from that single mRNA to get around the eukaryotic 1 mRNA, 1 open reading frame limitation is to make a polyprotein and then chop it up. That allows you to also make a capsid by sequential steps. So here we have poliovirus, where we're translating the precursor to the capsid protein, and this is composed of VP1, 2, 3, and 4, but it's made as a long precursor first, shown in the middle here, called P1, and then it folds into a folded P1, as you can see on the third panel from the left, VP1, 2, 3, and 4. And then finally, the viral protease clips the connections between these subunits to make the final structural unit. So now we've made a subassembly. This is going to go on, as you'll see in a few moments, to be used to assemble intact virus particles. All right, so so far we have assembly from individual proteins. Saw 2 examples of that. We have assembly from a polyprotein precursor. And again, these are examples. There are many other viruses that do similar things. The last example is chaperone-assisted assembly. And sometimes this is combined with some of the other mechanisms I've talked about. So here in panel C is looking at adenovirus assembly. We are making a hexon trimer. Hexons are those subunits of the capsid which are surrounded by 6 other subunits. right, as compared to the pentons, which are surrounded by 5 subunits. This is a hexon. It happens to be a trimer. And it's made simply by producing the individual proteins. This is one molecule of the subunit of the hexon protein 2, and then 3 of those come together to form that subunit. Subunit assembly, just like we've talked about, except in this case, the assembly of the monomer into trimers is assisted by a viral chaperone called the L4 protein. The name isn't important. The point is, a chaperone assists in assembly reactions. It helps assembly reactions to proceed error-free. There are also examples of cellular chaperones that participate in assembly. And on the bottom is now the same assembly pattern that we saw for poliovirus at the top, except I've added the cellular chaperone, which happens to be a protein called HSC70, heat shock protein 70, which has roles in the cell of quality control, making sure proteins are folded properly. But the viral proteins have a requirement for it to fold properly. So here, HSC70 binds to the P1 polyprotein as it's being produced, and it helps the P1 fold into the folded P1. So I showed it to you on the top without the chaperone, but actually it requires a chaperone. First HSC70, and then a second chaperone called P23, and then finally cleavage by the viral protease to give that last subunit. Okay, so these are just one of many examples of using chaperones. It could be viral or cellular to help the folding go properly. Let's take a look at the whole life cycle of poliovirus now to show you how this works. We've talked about how the virus attaches to receptors, how the receptor binding itself catalyzes RNA release into the cytoplasm. Talked about RNA replication separately, but today we're going to look at translation of the mRNA to a polypeptide. And remember, it's made into a polyprotein which is cleaved by virus proteinases. One of the subunits is the P1 precursor of the structural unit. We just saw how that folds and is cleaved. Here it is, the 5S structural unit. Now, how do you get a virus particle from that? 5 of these come together to form what we call pentamers. Makes perfect sense. 5 of the structural units come together. Now, if you could take those structural units and look at them, you would see a 5-fold axis of symmetry, and an axis around which there's simply 5 copies of that 5S structural unit. Now, we think the next thing that happens is the RNA, the viral RNA, binds to these 14S pentamers. They then assemble into what's called a provirion. So this is now a complete capsid with genomic RNA in it. It undergoes one final cleavage, and now we have infectious virus particles. So the 14S pentamers come together with RNA, of course, that's been produced by replication to form an intact particle. Now, you also see in this figure there's something called an empty capsid. If you look in infected cells, you can find lots of those because the 14S pentamers can also come together spontaneously to make an empty capsid without RNA. The making of a capsid is not RNA dependent. We think that those can come apart then, so the 14S pentamers later can be used to make particles with genomic RNA. So that's just one entire assembly reaction for poliovirus. Let's take a look at another example of assembly, and again using a chaperone. And this is happening in the nucleus. So this is a herpes virus, and again the assembly is sequential. Just like for polio, you make parts and you put them together in a series of steps. And we're going to distinguish that from another means of assembly in a moment. So remember that sequential assembly. Herpes virus replicates its DNA in the nucleus, and so it will assemble its particle in the nucleus as well. And is a very large, complicated capsid. So all the proteins that make up the capsid have to be made in the cytoplasm and imported. And some of them are shown here, all these pentamers and hexamers, triplexes, and one portal. You may remember that one of the axes of the particle is a portal through which the DNA comes out and through which, as you'll see, it goes in. So let's say that we've now got lots of DNA genomes in the nucleus. We've replicated them previously, and we've made them late in infection because of cascade regulation. And now you have to make a capsid. First, you make a procapsid, and it consists of the hexamers and pentamers Remember, this is a big particle, so we don't just have pentamers, we have pentamers and hexamers. And it's so big that it can't assemble on its own. It needs a scaffold. Okay? You know, when you make a building, you put a scaffold on the outside, and you can go up and work on it. And this is the opposite. The scaffold is on the inside. You make a scaffold, which is made of viral proteins. You can see them shown there, and you can trace where they're coming from on the left. And that holds the capsid together while it's being assembled. But of course, you don't want to leave the scaffold in there. You have to get the DNA in. So in the scaffold protein, there is built a protease. And once the scaffold is assembled and the capsid is done, the protease becomes active and it digests away the scaffold. So that's what you see in that second image with the little bits. That's what's left of the scaffold. After the viral protease, which is called VP24, gets rid of the scaffold. And then the last step, the genomic DNA comes in, and you can see it in blue there in the center, and the scaffolding proteins, what's left of them, goes out. We'll see how the DNA gets in a moment. But this is a very interesting assembly. It's sequential, of course, and you're making sub-assemblies. All these pentamers and hexamers, et cetera, are all sub-assemblies that have been made from individual polypeptides. You're packaging a protease to get rid of this scaffold. It's very interesting. The scaffold can be looked at as a chaperone to help assemble the particle, but you have to get rid of it, and that's how it's done. So this is sequential assembly. This one, poliovirus, they're examples where you make parts and you put them together in a series of events. That's what I mean by sequential assembly. Now here is a different kind of assembly, concerted. All right, because the particle forms at the very end and everything comes together. We put the RNA and the protein together at once and we get a particle. There's not a series of steps as there is for the other two viruses we talked about. So this is an example of influenza virus budding. We're going to talk about the different parts of this in more detail in a moment, but I just want to look at the very last points here. We've got viral glycoproteins in step 6. These are inserted into the plasma membrane. They've reached there by coming through the secretory pathway, ribosomes translating into the ER, then the proteins move to the Golgi and to the plasma membrane. At the same time, the genome has been replicating in the nucleus. It's associated with proteins, so it's a nucleocapsid. That comes out of the nucleus and goes to the plasma membrane. How it gets there, we'll talk about. What are the signals that allow it to get there? And then the virus particle forms by budding. The membrane pinches out, comes off, and now at step 7, we have a brand new influenza virus particle. So this is what we mean by concerted. The particle is formed in one step at the budding step. You got a genome, and you got the glycoproteins, and it buds out, and you have a virus particle. Okay, so we distinguish sequential and concerted examples of virus assembly. Let's take a closer look at the glycoproteins of influenza virus, just as an example. So on the upper left is the virus particle. We've just seen how it buds off from the plasma membrane. Remember, it's got an eight-piece segmented RNA genome. It's an enveloped particle, and there are glycoproteins in the envelope. And these will become really important to us when we talk about pathogenesis. So let's introduce them here. The hemagglutinin of the virus, the HA, and the neuraminidase. And there's also a couple of other proteins in here that are important. One of them is an ion channel called M2. We'll talk about that. We've actually talked a little bit about that during entry. We'll talk about it more later. All right, so these are glycoproteins embedded in the virus particle. The hemagglutinin is shown on the right. It is a trimer of 3 monomers, and the hemagglutinin is the protein that attaches to the cell receptor. We've talked about this briefly, and we're going to get into how this interaction, the attachment to cell receptors, is a big determinant of the pathogenesis of influenza virus infection. These proteins, these glycoproteins, HA in particular, are made first in the endoplasmic reticulum. The ribosomes sitting on the cytoplasmic face translate the mRNA. The protein goes right into the ER lumen, and then it remains attached to the membrane. And here is a schematic of the protein on the bottom. So here on the right is the membrane bilayer, and in red is the HA protein. So there's a little bit of amino acids on one side of the membrane, and then the majority of the protein, the ectodomain, is on the left here. And there are many interesting features of this protein. On the very end terminus, of course, is the signal sequence, which targets this protein to the endoplasmic reticulum, and that's then cleaved off so that the end terminus would start at the red sequence. These little Ys are additions of sugars. This is a glycoprotein, and these play an important role in its biology. The yellow lines are disulfide bonds. Right? You know what those are. They are linking cysteines together. And there are quite a few of them, as you see. And a very important part of this protein is the little green squiggly part called the fusion peptide. That is the peptide that inserts into the endosome membrane during fusion of influenza virus in the endosome at low pH. It's something we talked about a long time ago, and it'll come back when we talk about pathogenesis. This fusion peptide has to be exposed. And the way it's exposed is first by cleavage at that little arrowhead, and then at low pH, it folds up and inserts into the membrane. But you can see it's made embedded in the primary polypeptide initially. All right, so that is the flu HA. It's made, as I said, in the ER and shipped up to the plasma membrane. This is how that process works. An overview of maturation. HA0 is what we call the uncleaved influenza hemagglutinin. And at the top here, you can see the ribosomes. They're translating the HA mRNA, but they're bound to the ER, so the protein is going into the ER lumen. And of course, that happens because of the signal sequence on the protein. The protein is in red here. It folds up in the ER and oligomerizes, actually, early on in its synthesis. We're only showing one here for clarity. And then, as we've said before, vesicles pinch off of the ER. They bring these proteins to the Golgi, first the cis, then the medial, then the transcisterna. And at each step, there is a fusion event of the vesicle with the Golgi. Then another vesicle buds off and so forth and transports it through the Golgi. Through the Golgi, many important things happen. Further oligomerization, trimming of oligosaccharides, addition of specific sugars and so forth. And eventually, this protein reaches the plasma membrane. Now, in the next part of the course, we're going to talk about cleavage of the HA as an important determinant of pathogenesis. And right now, I want to just say that some strains of influenza virus, the HA is actually cleaved in the Golgi as it's coming out. And these strains tend to be, have a wider set of tissues that they infect than others because of that. We will come back to that because it turns out that most strains of flu are not cleaved. The HA is not cleaved in the Golgi. They're cleaved extracellularly. Okay, our first question. Sub-assemblies are involved in which of the following types of virus particle production? Concerted assembly, sequential assembly, assembly lines, chaperone-assisted assembly, all of the above. Wow, 75% only. I thought you would all get this. All of the above is the right answer. These are all, sub-assemblies are involved in all of these things. Concerted just means that you make the particle all at once. But remember, you make a nucleocapsid, that's a sub-assembly, and that goes to the membrane. None of you picked that, so I don't even have to explain it. Sequential assembly. Sure, sequential assembly is just another way of saying you make a sub-assembly, then add it to something else. Assembly lines have sub-assemblies as well. Remember the phage example where you made all the different parts? Those are sub-assemblies. Chaperone-assisted assembly, yeah, chaperone simply helps you to assemble the subunits, if you will, in an accurate manner. So all of these are correct. Sub-assemblies function everywhere. All right, let's now talk about genome packaging, this very interesting problem where you have to distinguish between viral genomes, which can be DNA or RNA, and cellular nucleic acids. You don't want to package cellular DNA or RNA. The answer is a packaging sequence or a packaging signal. And this is the sequence on the genome. Don't confuse it with the sequence on the protein because these signals, packaging signals, they're there to interact with structural components, right? The genome has some signal in it and interacts with a structural component, and that brings it into the particle. Packaging signal is on the nucleic acid, okay? So if I ask you in some form someday, what's a packaging signal, it's the nucleic acid. So that distinguishes the viral nucleic acids from the cell because they don't have packaging signals, so they are typically not incorporated into particles. So let's look at some examples of what is a packaging signal. First, we'll look at DNA viruses. Here are two examples, adenovirus and SV40. Adenovirus is at the top, and we're looking at the left end of the genome here. Here's the left end with its terminal repeat. There's the origin of replication. That's where the DNA polymerase binds with the terminal protein. And then we have a promoter here for an immediate early mRNA. And then there are a bunch of enhancers in here, which you may remember help get good transcription at that promoter on the right. And then down here with the little blue arrows, those are the packaging sequences. So there are lots of them, and they overlap. So you may say, how do you know that these are packaging sequences? Well, what people do is they make mutations along this part of the genome and see which ones prevent the incorporation of the viral DNA into the capsid. That's the operational definition of a packaging signal. Now for adenovirus, the way that works is these sequences bind a viral protein that happens to be called 4A2A. It's shown in the diagram at the lower right. So we have viral DNA is bound to 4A2, and that helps incorporate it into the capsid. So the DNA protein assembly is recognized by the capsid, and there we're showing on this right panel the DNA threading into the interior. So the assembly of AD is very much like that of herpes. You make the shell first, and then you put the DNA into it, and the way the DNA recognizes the capsid is by this packaging signal. So it's a DNA protein interaction. On the bottom is SV40, just to show you another packaging signal. And here, this part of SV40 should be recognizable to you. We have the origin of replication. There's only one of those in SV40 at the top of the circle, if you will. We have an early transcription unit on the right, and the late transcription would be to the left. Here's the packaging signal shown by the arrow here. These purple bars are binding sites for a transcription factor for SP1, and it overlaps with the packaging signal, which goes where the arrow indicates. And again, this allows binding of the DNA to a viral protein to help get it into the capsid. So these are not present on cellular DNAs. That's why they do not get incorporated into virus particles. Let's look at how herpes virus packages and gets the DNA in the particle. This will illustrate several principles, and it's just really cool how this happens. So remember, DNA replication of herpes virus. This is a linear double-stranded DNA genome, but when it infects a cell, the DNA is circularized by a host ligase, and then it replicates by rolling circle, and that's shown on the top there. Rolling circle simply means you make concatamers. You know what a concatamer is? If you have a genome of 100 base pairs, a concatamer would be multiple hundred base pair pieces all in one molecule. And for herpes, of course, the genome is much bigger. So you get these very, very long molecules with many, many genome equivalents in them. Now on the panel A here shows you the whole genome, the double-stranded DNA, and there are repeated sequences in it that we have just mentioned briefly. But now let's zoom in on the left-hand side of the genome on the bottom here, and you see these weird sequences called DR1, PAC1, DR2, PAC2, DR1. Well, these are the components of the packaging sequence for herpes, and PAC stands for packaging. What happens is when the DNAs are made as long concatamers, these PAC sequences, the PACs and the DR2, interact with a series of viral proteins. Let's look at the upper right here, right at the portal. So remember, there's one portal on a herpes virus particle. It's the way DNA gets out when the virus infects a cell, and when you're making new particles, it's how you put the genome into that preformed capsid. Remember, we've made the capsid with a scaffold. The packaging signals composed of DRs and PACs bind the portal together with a variety of cellular proteins. So that's how the DNA is recognized. Then the portal has associated with it a motor that winds the DNA through the portal, and that's shown happening in steps 2 and 3 and 4. There is actually an energy-dependent motor that is pulling this DNA in, so it's initially recognized by the packaging signal, and then it's pulled in. And when you get a head full of DNA, which is equivalent to one genome, that brings you to step 3. You now have 2 sets of packaging sequences, again, which are composed of these PACs and DRs, opposed to one another, and that's the signal to stop reeling in the DNA. There's a nuclease that cuts the DNA at that point, and now you have a head full of DNA in the capsid, and the remainder of the concatemer is outside, and that can go on to be taken in to another virus particle. So a couple of really neat concepts here. We have a portal through which, a very defined portal through which we're putting the DNA. We have these packaging signals that allow the DNA to be recognized by the portal, and then they tell the portal when you've got enough DNA in the head, so you never, in the capsid, so you never put in more than you need. Really, really clever mechanism of packaging, I think. So those are two examples of DNA packaging signals. Again, signals in the genome that interact with proteins that get the genome into the capsid. Let's look at RNA viruses. Two examples. One, retroviruses, where the packaging signals have been very well studied. Retroviruses, remember, have an RNA genome. It's a plus-stranded RNA. And we are looking at the left end of it here on the lower left. You can see PBS stands for primer binding site. That's where the tRNA binds to start reverse transcription. So we're very near the 5' end here. The packaging signal is shown by this arrow labeled psi, PSI, for P for packaging. And it consists of this RNA sequence shown above, which is not very long, but you can see it's highly structured, has lots of stem loops. And if you take this out of the viral RNA, it won't be packaged. And if you took that packaging signal and put it in another RNA, it wouldn't be enough to get that RNA packaged. There are some packaging signals that we can transfer to cellular RNAs if we want to make virus vectors to give people genes for gene therapy, which we'll talk about at the end of the course. You can put packaging sequences into them. But for the HIV packaging signal, it is necessary but not sufficient. There are other sequences we need for genome packaging as well. How does this work? Remember, the retroviruses incorporate 2 copies of the RNA genome into the particle. So the 2 RNAs both have this packaging sequence, and they interact with each other. And that's shown in the box above. And they make a structure lovingly called a kissing loop, because the 2 loops shown in red, so here's one of them, SL1, stem loop 1, interacts with stem loop 1 of the second RNA, and they form this kissing loop complex, which is shown at the top. The sequences that were formerly in this loop are now base pairing. So that's the dimer linkage. The 2 RNAs are in the virion held together by this base pairing. And that base paired sequence, in turn, is recognized by the viral nucleocapsid protein, or NP. NC is shown on the right here as part of the GAG precursor, matrix capsid nucleocapsid protein. We'll look at this in some detail in a moment. There's nucleocapsid. If we expand it, we see regions of the protein that are required for binding to this kissing loop RNA. So the RNA does not have a binding site for the nucleocapsid unless there are 2 of them present, and then they make a structure that the nucleocapsid can bind to. And the nucleocapsid, as you will see in a few moments, is part of a protein that goes to the plasma membrane, and that's how it gets incorporated into particles. So we have to get to that point before you see the denouement of this, but the point here is that this is the RNA packaging signal that's interacting with nucleocapsid and pulling it into the budding virus particle. All right, so that is pretty straightforward. Many RNA viruses have these kinds of signals, and they've been identified, but a really interesting conundrum is what happens when you have more than one segment in a virus. How does that work? People have been trying to figure this out for many years. So here's influenza virus, which has this 8 RNA segments in the particle. And you have to have 8, and you have to have the right 8. You can't have 7, it won't be infectious. You can't have extra 1 or 2. You've got to have 8 unique RNA segments. Now, it turns out that if you have a cell in which the virus is replicating, you have a pool of all 8 genome segments, they're all replicating. If you just randomly grab 8 segments from the pool, you'll be right 1 in 400 times. You'll grab the right 8 segments 1 in 400 times, and that's more or less what the particle to PFU ratio is for flu, about 1 in 400, 400 to 1, particle to PFU. So a random mechanism could explain how you get 8 of the right segments in the particle. It's 399 times you get it wrong, and those particles are not infectious. But it's too simple. So people kept looking for something else, and there's now evidence for a specific packaging sequence on each of these viral RNAs. Let me show that to you here. So here on the upper right is a diagram of the RNAs of each segment. From top to bottom, there are 8 segments here, and each encodes 1 or more proteins. And the yellow part is the open reading frame that encodes the protein, and the blue parts are what we think are the packaging sequence. So each segment has a unique packaging sequence. The idea being that this is needed to make sure that you get all 8 segments into the particle. We actually don't know how they interact, but in one experiment, they swapped the packaging signals of HA with the packaging signals of the NS, 2 separate genome fragments, and they got incorporated perfectly well. So these sequences can work on different segments. The way we think this happens is on the upper left. This is a diagram of a budding influenza virus. You can see we're just showing 1, 2, 3, 4, 5, 6 segments here for clarity. But the RNAs are aligned in parallel, and they're all budding out perpendicular to the membrane. And if you take an EM of virus particles, you can actually see the RNA segments in them as little dots, showing you that they're oriented in a parallel fashion. So somehow these sequences are interacting with each other and probably also with viral proteins so that you get the right 8 segments in each particle. You always get 8 segments. Now, there is a bacteriophage where it's quite clear that we can have selective packaging, and this is shown here. It's a bacteriophage Phe6. Its genome is 3 double-stranded RNA segments, and this is very much like a retrovirus, an animal virus, which has many more double-stranded RNA segments. And these viruses, just like the retroviruses, are made up of 2 protein shells, an outer capsid and an inner capsid, and in the very interior are the 3 genome segments, the large one, the medium-sized one, and the small one, SML. So it turns out that there is a serial dependence of packaging. These RNAs have sequences in them that say the S is always the first to get into the particle. It is then followed by the M. If there is no S, the M will not go in. And then once the M is in, the L goes in. So the order is SML, and it doesn't work any other way. So presumably the signals on the RNAs dictate that. And so it could be similar for influenza viruses. This virus, interestingly, has a particle to PFU ratio of about 1. And remember, I said flu is about 400. So flu makes a lot of mistakes, maybe because it's randomly packaging. And this guy isn't randomly packaging. It has got a serially dependent mechanism for getting exactly the right segments in each particle. So maybe that's why the particle to PFU ratio is 1. So I think this is a very neat example of how serial packaging could really help your particle to PFU ratio. All right, our next question. Packaging signals on viral blank interact with viral blank during virus assembly. Packaging signals. A, lipids, proteins. B, proteins, sub-assemblies. Genomes, proteins. Proteases, membranes. Proteins, genomes. Okay, 92%. Packaging signals on viral genomes interact with viral proteins during virus assembly. That's why I repeated so many times earlier that a packaging signal is on the viral genome. But some of you, 8% of you, feel that packaging signals are on viral proteins. No, packaging signals are on viral genomes. Okay, now you should not ever get that wrong again. Let's talk a little bit about budding, that last step in making an enveloped virus particle. We have to acquire an envelope. We often call this membrane from the cell an envelope. And most enveloped viruses, this happens after assembly of the internal structures. Now remember with influenza, and we'll see that again, you make the nucleocapsid, and then the particle forms as it buds out. That is after assembly of internal structures. But for some viruses, the whole thing comes together at the stage of budding, simultaneously with assembly of internal structures. Retroviruses do it that way. It's a bit different. And we'll look at both in a moment. But on the bottom is different determinants of budding in different viruses. It turns out that in some cases, only one viral protein is enough to form a bud. So for example, on the left, there are four different examples here. On the left, the glycoproteins and the capsid are needed for budding. You have to produce both. If you just produce one or the other, you don't get a bud form, and those drive formation of the budding particle. Example 2, the internal matrix or capsid protein can drive budding. So as we'll see in a moment, the matrix protein is a viral structural protein present just below the membrane in many of these enveloped viruses, and we think it gives structural integrity to the particle. So we call that a matrix protein, or M protein. It's shown in blue. So that, for some viruses, can drive budding. Example 3, the envelope proteins alone are enough to drive budding. Now you may be saying, why do I care if the envelope proteins are enough? Well, it turns out that we can make vaccines by just producing the HA of influenza virus in certain cells. You can do this in plants, as we'll see. You can make flu HA in plant cells, and it will make particles, and those vaccines are currently being tested. And it doesn't work for every virus, but when it does, you're lucky, because you basically get a lipid vesicle with an HA in it, no infectious genome, and that can be very immunogenic. And finally, the last example, the matrix proteins drive budding, but sometimes you need ribonucleoprotein there as well. So the point here is that the budding process is driven by viral proteins, different combinations of them. But as you will see, what is going on here is the viral proteins are hijacking a cellular process. So let's look at influenza virus budding. And let's take an overview of the whole cycle. The viral genomes replicate in the nucleus. So here, a lot of things going on. On the left, we're making mRNAs from the input RNAs that have come in by infection. Those go out into the cytoplasm, of course, and various proteins are made. The membrane glycoproteins get made in the ER and get shipped up to the plasma membrane by the secretory pathway. We've talked about that. We have to make new nucleocapsids in the nucleus, so we have to replicate the plus strand through a plus strand intermediate to get more minus strands. Then we coat them with nucleocapsid protein, and of course that has to be made in the cytoplasm and shipped in. And then we also need to attach a matrix protein, the blue protein just below the membrane, which you can't see here. That's called M1. That's made in the cytoplasm, goes in the nucleus, and binds to this nucleocapsid. So now this sub-assembly, RNA and a bunch of proteins, goes out of the nuclear pore and goes up to the plasma membrane. And of course, it travels along a microtubule. That's why I put in these brown tracks here to show you and remind you again, it doesn't diffuse up. It moves very specifically with motor proteins on those microtubules. Then this RNP assembly goes up to the plasma membrane. How it does that, you'll see in a moment, and that stimulates the production of a budded particle. This is concerted assembly. The particle forms all in one step. Let's take a look at the M1 protein of influenza virus, because it turns out it's the M1 protein that allows the ribonucleoprotein, the RNA, plus other proteins, including the polymerase, to get up to the plasma membrane and stick there. This is a line diagram of the M1 protein, and you can see interesting regions. For example, on the left, hydrophobic regions. That's what you need to bind to lipids. There are a couple of ways that a protein can be targeted to membranes. One of them is a signal sequence, obviously. Another one is a hydrophobic region, and we'll see another one in a moment. So this is the region that allows the M protein to embed or get near to the plasma membrane. And just while we're looking at this protein, look, there is a nuclear localization signal. M1 is made in the cytoplasm, so it's got to get into the nucleus. That's what the NLS does. And upstream of it, there's an NES. It's not Nintendo Entertainment Systems. This is nuclear export sequence. Gets the protein out of the nucleus. If you look, let's go back one slide. I'll show you that. Here's M1 bound to the RNP. M1 got in by a nuclear import signal, NLS, and now it's going to get out by an NES, and so it can go up to the plasma membrane. So that protein's got a lot of functional regions, and that's how it gets up here to the plasma membrane. Just for comparison, VSV also has an M protein below the membrane of those bullet-shaped particles, and the M is shown here. And you can see a hydrophobic region and a positively charged region. The hydrophobic region is for membrane binding. The plus region is to bind to the RNA, which is negatively charged. And you can, how do you know this, you may say? Well, you take these parts out of the proteins and you see what functions are affected. Let's go to retroviruses now. This is different. Here, the particle doesn't form until late, as opposed to influenza virus, where the budded particle forms coincident with the addition of the nucleocapsid. Now, if we think back to the reverse transcription lecture, the genomes are made in the nucleus from the provirus that's integrated. They come out into the cytoplasm, and these are unspliced messages. Remember, we have to get a way of getting unspliced messages out. And there's the dimer linkage. So we have two genomes in green interacting via that packaging sequence. We have the dimer linkage here, and that allows the nucleocapsid protein to bind to the RNA. Where's the nucleocapsid? On the right. Here is our GAG structural precursor. This is the GAG protein. Matrix, capsid, nucleocapsid. The nucleocapsid, so this thing is made as a precursor, but it's not cleaved yet. The cleavage happens later. It's made as a precursor, and the nucleocapsid, which is embedded in the precursor, let's follow this arrow and go up here where it's labeled GAG. You see matrix, capsid, nucleocapsid, and a couple of small proteins. They're all linked together still. This little protein assembly here binds the RNA. The matrix protein has signals to target it to the plasma membrane. So this whole RNA protein assembly goes up to the plasma membrane, where, of course, the viral glycoproteins are already inserted. They're called TMSU. So now we have two RNAs brought up here by virtue of their association with nucleocapsid, which are the blue spheres. We have a layer of capsid protein forming in red, let's say, or brown. And then we have the matrix protein in blue. It starts to bud out. Now, at the same time, we have to get a longer precursor that has our enzymes, the reverse transcriptase and the integrase. That's called GAG-POL. Remember, frame shifting or translational suppression sometimes gives you a longer precursor. And that way, you can bring in reverse transcriptase, integrase, and protease into the particle as well. Not as frequently as the other one. You just need a few of these, 50 or so RTs per particle. But you can see without RNA, they don't need any RNA. They slot right in to the growing particle. And the RT, for example, is light blue. And you can see you've got a molecule of RT there. And this proceeds in step 2. The bud forms, and the whole structure moves out. And we still haven't made a complete particle yet. And now we have a free particle that's budded off from the cell. But it's very different from the final particle. The capsid still is lining the membrane, and you see there's been a transformation already from when the particle has budded off. These nucleocapsid proteins, which are originally attached to the capsid, are now coating the RNA. So they've moved locations. That's the action of a protease. And then the final maturation step, the capsid subunits are released from the M protein, and they form an icosahedral capsid surrounding the RNA. So this is an example of a particle that doesn't form at the point of budding. It actually matures afterwards. But the really cool part here is how the RNA is brought in by this precursor protein. NC is embedded in the precursor, and M has similar signals to the other M proteins that we've seen to get it to the plasma membrane. So let's take a close look at the GAG protein of retroviruses. Here's the same cycle that I just showed you at the upper left. So the matrix is the blue protein. Unfortunately, it's colored differently here, which is too bad, but it should be blue. And that's the protein that will bring the whole assembly to the plasma membrane. Look, at its N-terminus, it has myristate. That's a fatty acid. That's another way you can get proteins to membranes. And it also, if you look closer, has positively charged hydrophobic regions. So these all help target matrix to the membrane and bring up the RNA that's bound to the nucleocapsid in the same protein precursor. So So you see how having a precursor is dragging everything into the particle, and then when the particle's made, they'll all be cleaved. And this is just to remind you that nucleocapsid has binding sites for specific RNAs. So lipid is the other way to target proteins to membranes. We talked about signal sequences and hydrophobic patches. You could actually covalently link a lipid moiety onto protein. And this one is myristate. There's the lipid there, and it's typically covalently linked to a protein that needs to be targeted to a membrane. And this is one of several different kinds of lipids that can be added to viral proteins. And the way this works, the protein's made, and then the lipid is added afterwards. It is covalently attached, and you saw the example at the N-terminus of matrix. The addition of a lipid targets the protein to the plasma membrane. Okay, a last interesting point about budding. A number of years ago, a number of laboratories were going through the retroviral GAG protein and making amino acid changes to figure out what parts of it did what. You know, what parts bound RNA, what parts bound the plasma membrane. And one set of changes, here's the GAG precursor for HIV and a mouse retrovirus. You can see matrix, capsid, nucleocapsid. When they got to this little protein at the end called P6, or P12 for the other virus, when they made changes in that, what they saw in infected cells, the viruses would bud off, but they would remain attached to the plasma membrane by these stalks. They would never pinch off. They had a really interesting phenotype. And these were called late domains because they arrest budding at a late stage in viral replication. That's what they're called now, L domains of these proteins. And since then, they were found in all plus and minus strand enveloped viruses. The glycoproteins have, or other structural proteins have these late domains. If you mess with them, you get particles that never leave the cell. It turns out that those late domain viral proteins interact with the host cell machinery, which is diagrammed here. It is the ESCRT pathway. It is a pathway in the cell, as we'll see in a moment, that's normally used to do membrane scission events like those involved in budding off pinching off of a budded virus. And why changes in P6 and P12 affect budding is because these proteins, through a very specific amino acid sequence, interact with components of the ESCRT pathway, including proteins called TSG101, ELIX, and so forth. So these small proteins, P6, P12, which nobody had paid any attention to, are now crucial for pulling these particles into a membrane scission pathway. So the endosomal sorting complex is required for transport, or ESCRT. It's a machinery originally discovered in yeast, now known to be present in all eukaryotic cells. It is responsible for things like pinching off two cells when they divide. You have to have that last event to break the two membranes. That's called abscission. The ESCRT proteins are involved in that. During the formation of exosomes, these are little vesicles that are released from cells. These little vesicles are made by virtue of the ESCRT pathway, the formation of multivesicular bodies. Endosomes coming into the cell fuse with a larger vesicle. You can see one fusing right there. All of these reactions are carried out by the ESCRT pathway. There are a series of proteins that are involved in membrane fusion and abscission. You can see some of them here, ESCRT3, ESCRT1. Now, normally, this doesn't happen at the surface of the cell, except the virus has absconded with the ESCRT pathway. So viral proteins, those proteins with the late domains, bind components of the ESCRT pathway, and they drive budding via these mechanisms that would otherwise be involved in making various vesicles in the cells. So the bottom line is that this and many other viruses, this happens to be a retrovirus, which we show here, have hijacked the ESCRT pathway and now divert it to the plasma membrane. How do they do that? Well, the M protein goes up there and brings the component of the ESCRT pathway with it and makes it bud out rather than forming vesicles in a cell. It's really brilliant, as far as a virus could be brilliant. All right, to end up our discussion of budding, budding can happen at every membrane in the cell. It can happen at the nuclear membrane for herpes. It can happen within the ER, it can happen within the Golgi, and of course it can happen at the plasma membrane, as we've seen throughout our discussion. So remember that any membrane is fair game for budding of virus particles. Now you may be wondering, how the heck, if you bud into the nuclear membrane, how do you get out of the cell? Well, I'll show you one example. Let me show you an example of herpes which buds from the nuclear membrane, and then you could use this to figure out how budding from all the other sites works as well. But herpes capsids are assembled in the nucleus full of DNA. Then they bud out of the nucleus into the ER. So there's a herpes virus particle in the ER. But that doesn't do herpes any good. It's got to get out of the cell, right? What does it do? It fuses with the ER membrane and loses the membrane it acquired at the nucleus. So now it's in the cytoplasm, and it's essentially a naked particle surrounded by some viral proteins, but there's no membrane. Next step, it buds, or it fuses, no, it can't fuse, it buds into the trans-Golgi network and acquires a membrane there. Now then, instead of fusing again to get out, it acquires a second membrane by budding, so a second budding event in the trans-Golgi. So now you have a normal herpes virus with a capsid and a membrane, and it's got another membrane around it. That goes up to the plasma membrane, the outer membrane fuses, and the particle is released. So that's how you bud out of the nuclear membrane and keep the viral membrane. And an interesting exercise would be to figure out where that viral membrane comes from. So you could use that to figure out where all of those, how all those viruses who bud from the Golgi or the ER or the plasma membrane acquire their membranes. Now, I've talked a lot about how particles of viruses are moved on microtubule networks. And here's an example with vaccinia virus. So these virus particles form in the cytoplasm, and they're moving here along microtubules on motor proteins towards the plasma membrane. They happen to have two membranes on them. One of them fuses, so now you have an extracellular virus particle here called CEV. And then a very cool thing happens, because this is just sitting on the plasma membrane. This particle binds a receptor labeled X here, which stimulates a signaling pathway through a kinase called SRC that triggers actin polymerization. Normally actin's fibers are lying parallel to the plasma membrane just beneath it. This signaling causes the actin filaments to polymerize, and they make these long strands which basically propel the virus off of the cell surface into another cell. A great way of spreading. And here on the upper left is a fluorescent micrograph of some of these actin projections. So there's the infected cell, and these are the actin projections. And in red are the virus particles at the tips of the actin, which is what's diagrammed below. And on the upper right, you can see a virus particle in black, and below it is this actin tail that's been formed by this signaling pathway. So it's a very cool way to get out of the cell. Now, most of these virus particles that bud from the surface, they just float away, and we'll talk a little bit about that later. But vaccinia virus remains stuck to the surface, so it utilizes the actin network to push it. And what this does, it pushes it into the neighboring cell, so those particles can infect those cells as well. The last thing I want to just address is we've talked a lot about how envelope particles get away from cells, right? They eventually bud from the plasma membrane and are released as particles or pushed off in the case of vaccinia. What about naked particles without a membrane? How do they get out? Well, many of these viruses kill cells. Here's a series of photos which we've seen before of poliovirus killing cells in culture. It lyses the cells, and that's how the particles get out. Lysis is a combination of many effects of the virus on the cell metabolism. These viruses induce cell death of various types. Viral proteins like viroporins actually poke holes in cell membranes to allow the viruses get out. And the general inhibition of macromolecular processes in cells like DNA and RNA and protein synthesis, this all leads to a loss of membrane integrity, and the viruses get out. I would say for the majority of non-envelope viruses, they get out by lysing the cell. But there's a growing body of evidence, and it's quite interesting, that there are non-lytic mechanisms for release of these non-enveloped viruses. And they are diagrammed here. For example, The picornaviruses and many RNA viruses, I told you, induce the formation of vesicles, membranous vesicles in the cytoplasm, and the RNA genome is replicated on the surface of those vesicles. Late in infection, it's been shown that some of these vesicles can actually, when they form, incorporate virus particles, eventually move to the plasma membrane where they fuse and release virus particles without lysing the cell. And if you've heard of the membranous vesicles called exosomes, these are part of normal uninfected cells. They are generated from within multivesicular bodies. They often are used to transport molecules of various sorts to other cells, including proteins and small RNAs, microRNAs. Some viruses actually get packaged into exosomes and are released this way. So here, and in fact, we had this on our last exam, the observation is that hepatitis A virus, which is a naked picornavirus, sometimes acquires an envelope like this. And that is by this pathway. Without lysing the cells, these viruses can get out. So just remember, envelope viruses are released as the bud forms at the plasma membrane. Lytic viruses typically are released by cell lysis, but in some cases, these non-lytic mechanisms may be in effect.